Dementia, and cognitive fragility in general, are among the most significant threats to healthy aging. Recent studies have shown that 1 in 4 people in the US, 80 years and older, suffer from dementia. Researchers have linked this loss of cognitive ability to change in the mechanical structure of the brain. In this work, we review these age-induced changes, first by looking at the brain as a whole, and then gradually moving down to the cellular level. After the age of 40, the entire brain begins to undergo significant macroscopic changes. The brain shrinks in size and becomes softer and more malleable. Compared to that of a 40-year-old, the brain of an 80-year-old is 19% smaller and 28% less stiff. These changes disrupt the folding of the cortex, with different brain regions becoming either more or less folded. Another subtle change with age is a disruption to cerebral blood flow. Brain arteries become stiff, and the microvessel density decreases. These areas of decreased blood flow seem to have an increased risk of white matter lesions, and these can disrupt the normal cognitive functioning. At the cellular level, neuronal loss and morphological changes in neurons, especially in the structure of dendrites, have been linked to cognitive impairments and behavioral changes in aging. As the brain ages, dendrites begin to exhibit shorter shafts and fewer spines and branches, which largely influence signaling mechanisms and synapse activity crucial to learning and information processing. Diving deeper into cellular mechanisms also shows that cell senescence contributes to brain aging. Senescent cells are cells that cease division but continue activity and secretory function in the body. Astrocytes, which are glial cells that support neurons and central nervous system activity, experience senescence that affects memory-associated tasks in the hippocampus. Another microscale phenomenon that influences cognitive decline is the expression of toxic proteins, like amyloid beta, linked to Alzheimer's development. These proteins contribute to mitochondrial dysfunction, which plays a key role in declining cell function and aging. This contributes to amyloid beta accumulation that further worsens mitochondrial function, which can lead to neural degeneration and dementia. Our review has found that many of these aging mechanisms influence each other, and that large-scale manifestations have causes rooted at the cellular level. The understanding of these mechanical changes may have a profound impact on diagnostics, with the in vivo identification of certain mechanical changes potentially predictive of disease states. Certain mechanical changes may also provide targets for the next generation of therapeutic drugs which attempt to slow or even completely halt cognitive decline.